All right, so let's carry on looking at microorganisms as an important tool of biotechnology. So we said that the first advantage of using them is that they're really easy to grow. So bacteria in particular reproduce really fast in a process called binary fission, and we're going to look at that in a minute on a different slide. But basically, it's cell division. E. coli divides every 20 minutes, yeah, under optimal conditions, obviously. So if it's got enough nutrients and so on, every 20 minutes. So that means that they reproduce really, really fast. Single-celled fungi are a little bit slower, but they also reproduce relatively fast. And both of these, so yeast and bacteria, can be grown in really large fermenters, and that's nothing else than a big recipient. And we're going to look at what a biofermenter looks like as well. They usually simply need like a good nutrient medium, and that can be easily produced at large scale. So that makes it really easy to grow them. Yeah, so on the picture, you see what an industrial biotech lab would look like. Yeah, so you've got a big fermenter, and then you've got some machines steering that, and there's a couple more fermenters at the back. But this is really what you need to grow bacteria or yeast. Yeah, binary fission, very, very easy. Yeah, so bacteria have no nucleus. So therefore, they don't need to do mitosis. Remember mitosis? You've got this really complicated dance of the chromosome. They got to, arrange, and then they're being pulled into two different directions towards the poles of the cell. Here, things are far easier because there's only a single chromosome. Yeah, so you've got a very simple picture of a bacterial cell here, which shows the cytoplasm and the chromosome, the bacterial chromosome. Then the DNA is replicated, so then you've got two chromosomes, and cytokinesis happens, and then in the end you've got two identical daughter cells. So there are, of course, similarities with mitosis, because you're getting the two identical daughter cells, which you do as well in mitosis. Hopefully you remember that. But the process is far simpler. There is no nucleus, there is no nuclear membrane, there is just a single chromosome. It's basically really, really straightforward, and bacteria can deal with it very, very fast. Because they are small, their DNA is small, they can run through the process really fast, and that means that they will grow very well. So if this binary fission is successful, then how do microorganisms grow? Well, I'm showing you a growth curve here. So on the x-axis, you've got the time in hours, and on the y-axis, you've got the number of bacterial cells. So it's really important to realize that when we talk about bacterial growth, obviously they're not growing in size. Yeah, so it doesn't really respond to your definition that you've learned about the 7 processes of life, where growth is a permanent increase in size. That is not the case for when we talk about bacterial growth, so it's not really proper growth in that sense. What it simply means is that there is a lot of cell division, and the density of cells will increase, so the amount of cells in the medium will increase. That's what we mean by bacterial growth. So in the first phase, from 0 to roughly 2 hours, this is all under optimal conditions, obviously. The bacteria won't do very much. They're sort of settling in their new nutrient medium. They're getting their bearings, if you like. Well, what really happens is not that they're getting their bearings, but they are adapting their metabolism to the nutrients that they find in the nutrient medium. So for instance, if they find glucose, they need to adapt their metabolism to digest glucose. And if they find something else, well, then they'll adapt their metabolism to be able to digest something else. Once their metabolism is adapted to the nutrients, the bacteria will go into what we call the exponential phase. And you will hopefully remember that from when we talked about human populations, because it really is very similar. Yeah, so when the conditions are right, populations go into, and nutrients and resources are unlimited for the time being, then populations go into exponential growth. Yeah, so we call this the exponential phase. You may remember that when we talked about human populations, I also mentioned the word log phase. So after the lag phase, now we've got the log phase, where, in this case, let's go back to the cells. So they divide, binary fission, and their population numbers explode. And well, what happens? Well, they're in this enclosed space, which is the biofermenter. And at some point, they don't have enough nutrients to just keep exploding in size. So they will be, there is a slowing down in growth, and they will be entering what we call the stationary phase. During the stationary phase, there is competition. Yeah, so you have to imagine that there is a limited amount of resources, a limited amount of nutrients, and so that limits, of course, the further cell division. There is competition between the different bacterial cells, and so now they are in stationary phase. During stationary phase, they may also produce what we call secondary metabolites. What are these? Well, there are substances which, help them get the edge over the competition. So, for instance, they may be producing an antibiotic to ward off other cells or something like that. Yeah, so that's when they start making substances to defend themselves against the competition, because you have realized now they don't have, there's no unlimited availability of nutrients, so they are all trying to get the edge over one another. But finally, that is not enough, because really what is lacking is nutrients, and at some point they will start to die. So in the death phase, these nutrients are used up, but also toxic products can build up, because bacterial cells or yeasts can produce chemicals that are toxic to themselves, and that means that their life is limited by that. So for instance, yeasts produce alcohol. Well, at some point, the alcohol content in a beer, for instance, is so high that the yeast cannot survive. Yeah, so this is sort of a feedback, and this is why you can't get an unlimited amount of alcohol in a beer, because at some point the yeasts that are producing it will die. Okay, so in the death phase, as the name promises, cells die. Now let's have a look at the biofermenter, as I promised you we would. So biofermenter, and there's a worksheet on that which I would like you to fill in and stick in your exercise books. A biofermenter, obviously there's lots of models available, but there will always be the same basic components. Yeah, so starting at the top left, we have inlets where the microbes get into the fermenter, where the air gets into the fermenter, and where nutrients get into the fermenter. Then top right, we have a gas outlet. Yeah, so any gases that they produce, we need an outlet, because otherwise the pressure will increase inside the fermenter, and we don't want that. So we have a gas outlet. At the very top, we have a stirrer. At the very top, I mean, you've got sort of the top part of the stirrer, if that makes any sense. And so this is sort of a long rod with paddles on it. Yeah, and it says paddle stirrers, and basically that thing rotates, and so it'll mix the content of the fermenter. So the orange stuff will be the nutrient broth, and the bacteria live in that. And obviously they need air as well. They need oxygen, most of them, not all of them. And like I said, that is going in through the top left inlet. Okay, so what else do we need? We always have a heating or a cooling system. Yeah, you need to be able to adjust the temperature that your microbes live under, and that is achieved through this heating or cooling system. So water in, water out. The water might heat the whole thing, or the water might cool things down, depending. Then you've got a computer with a couple of probes. Now the probes, they basically determine where you are, at what stage you are. How much of your product have you already got, or how much of the input have you already used up. If you're giving them nutrients, how many of these nutrients are left, do you have to top it up, etc. So that is all linked up to a computer, which will tell you exactly how your culture is doing and what they may eventually need. And then at the bottom of the vessel of the fermenter, there is an outlet, and that is the outlet for the product. So basically you open that up and you fill your bucket with some product, and that's almost as easy as it is. Obviously you have dead cells in there as well, and you may have other products in there as well, so you might have to do some you may have to do some separation, use some separation methods to purify your product, but other than that, it's really relatively straightforward. Yeah, so please make sure that you all fill in your labels on the worksheet that I give you, and that you stick that into your exercise book. Yeah, so you have to know the different components of a biofermenter. Okay, so here's an application using enzymes from microorganisms. So what's an enzyme again? I ask you? Obviously you can't answer, but hopefully you all remember. So if you don't remember, maybe put on pause and take some time to remember. Yeah, I know you're not going to do that. But in any case, hopefully you will remember some of this. So an enzyme is a biocatalyst. So that's a complex organic molecule, and that's a protein. Yeah, so it's a special type of protein which speeds up the rate of a specific reaction. Hopefully you remember that. That's an enzyme. Enzymes are specific to their substrate and also the type of reaction that the substrate will undergo. So for instance, an enzyme that catalyzes the breakdown of lactose to glucose and galactose will only do that. Its only substrate will be lactose, and the only reaction that it'll be performing on the lactose is to break it down into galactose and glucose, nothing else. Now the enzyme itself doesn't get involved in these chemical reactions, and it can be used over and over again. And in biotechnology, that's really great. That's something that you want. Yeah, so now basically you're going to be growing your microorganisms to get a specific enzyme from them. Yeah, so remember the biofermenter that we let out the product, your enzyme is among these products. And that enzyme you're now going to use. What can you use it for? Well, the sort of model application that they present to you in the book is the digestion of lactose in milk. Yeah, so milk contains lactose, which is a disaccharide. Hopefully everybody remembers what a disaccharide is. Contains 2 sugar units, in this case glucose and galactose. Yeah, if it contains glucose and galactose, it's called lactose. If it's 2 glucoses, it's called maltose, so that's something entirely different. Now the enzyme lactase breaks lactose down into its components. Yeah, so be careful. There's lactase, the enzyme lactose, the sugar. But it turns out that lots of people lack this enzyme. Now we all have it when we're born because we need it to digest the lactose in the milk that we get from our mothers. But by the time we turn 6 or 7, we usually, you may say, far earlier than that, but this is, biologically when children stop drinking their mother's milk. And so this is the time at which we do not produce lactase anymore. Now, the cow's milk that we are drinking contains lactose. See, I'm getting confused myself. So remember lactose, the sugar, lactase, the enzyme. The cow's milk that we're drinking contains lactose, and many people are tolerant to lactose. That means that they have the enzyme lactase beyond the age of 6 or 7. But the natural state, and in many populations it's left at this natural state, this enzyme is not working anymore after that age, and so drinking milk with lactose will give you sort of tummy ache and indigestion. So lactose-free milk is a good thing. How do we get it? Well, basically we obtain lactase from microorganisms. So certain bacteria, for instance, will produce it for you. And then you take this lactase and fix it onto small beads. Yeah, so as you can see on the picture on the right, these green beads, they're all coated with lactase. So there's lots of lactase molecules sticking to the surface of these beads. You can't see them because they're too small, but they're there. Then you take the milk and make it flow past these beads. Now, the time that the milk takes to flow past the beads is enough for the lactase fixed on the beads to break down the lactose flowing past. And so your product will be lactose-free milk. Okay, so here as well, I've got a worksheet for you that belongs to the slide. So please use the slide to complete the worksheet.